Hey students, in this video we will discuss about blood, its component and its containing and its structures, functions and how it regulates our body. Introduction. Blood is a connective tissue in fluid form, fluid of life, fluid of growth, fluid of health. Properties of blood. Its color, we will discuss about its color, its volume, reaction or pH, specific gravity or viscosity. Composition of blood. Blood contains the blood cells which are called formed element and the liquid portion known as plasma. Blood cell. Red blood cells or erythrocytes, white blood cells are also known as leukocytes. Platelets is also known as thrombocytes. चलिए हम पढ़ते हैं आरबीसीज के बारे में. रेड ब्लड सेल आल्सो नोन एज red blood corpuscles or erythrocytes. Erythrocytes have the biconcave shape with a movement. Red blood cells are bigger in size. Red blood cells of the red blood cell is coming from hemoglobin. Red color, the जो रेड ब्लड सेल का कलर होता है लाल कलर का, वो हीमोग्लोबिन की वजह से होता है. हीमोग्लोबिन इज़ मेड फ्रॉम आ हीमाटीन डेट कंसिस्ट ऑफ आयरन एंड प्रोटीन. हीमोग्लोबिन है वो मुश्तमिल होती है हीमाटीन डेट कंसिस्ट ऑफ आयरन एंड प्रोटीन. हीम एंड ग्लोबिन, इट इज़ टू एलिमेंट. हीम मीन आयरन एंड प्रोटीन मीन ग्लोबुलिन. ऑक्सी हीमोग्लोबिन इज़ formed by the oxygen and hemoglobin in red blood cell. WBCs. There is larger than red blood cells. However, the amount of white blood cell is low. This is also known as leukocytes. One white blood cell is 600 red blood cells. White blood cells are colorless and have nucleus. 1 ml blood is equal to 4,000 say 11,000 white blood cells. Type of white blood cell. वाइट ब्लड सेल्स की टोटल 2 टाइप होती हैं, ग्रेन्युलोसाइट्स एंड ए ग्रेन्युलोसाइट्स. जो ग्रेन्युलोसाइट्स के अंदर आते हैं वो होते हैं न्यूट्रोफिल्स एंड इयोसिनोफिल्स, और ए ग्रेन्युलोसाइट्स के अंदर आते हैं मोनोसाइट्स और लिम्फोसाइट्स. ऑन द अदर हैंड ग्रेन्युलोसाइट्स के अंदर आते हैं न्यूट्रोफिल्स, इयोसिनोफिल, बेसोफिल्स. लेकिन ए ग्रेन्युलोसाइट्स के अंदर आते हैं मोनोसाइट्स एंड लिम्फोसाइट्स. प्रोड्यूस एंटी क्लोटिंग प्रोटीन called heparin. Produce chemical to stimulate the tissue damage repair called histamine. These are produced from granulocytes. और एक granulocytes प्रोड्यूस करते हैं डिफेक्ट बैक्टीरिया को ख़त्म करते हैं. प्रोड्यूस ऑफ एंटीबॉडीज़ को प्रोड्यूस करवाते हैं उसकी प्रोडक्शन के बाद. लेट्स टॉक अबाउट प्लेटलेट क्या होते हैं, ये क्यों होते हैं, मतलब कि ये क्या काम करते हैं हमारी बॉडी में, चले पड़ते हैं. प्लेटलेट आर वेरी स्मॉल एंड हैव नो न्यूक्लियाई. प्लेटलेट बहुत छोटे पार्टिकल होते हैं और ये न्यूक्लियाई नहीं रखते हैं. दीज सेल्स हैव ग्रेन्यूल्स, ये ग्रेन्यूल्स रखते हैं. व्हेन यू आर वाउंड, जब आपको कहीं भी चोट लगती है, ब्लड विल नॉट कम आउट ऑफ द वाउंड कंटिन्यूसली. खून लगातार नहीं निकलता रहता, क्योंकि ड्यू टू ब्लड क्लोटिंग प्रोसेस, क्योंकि ब्लड जम जाता है. एंड इट इज़ क्विकन बाय प्लेटलेट. तो ये सारा प्रोसेस होता है प्लेटलेट की वजह से. लेट्स टॉक अबाउट कंपोजिशन ऑफ प्लाज्मा. Solid 7 to 8% and it's contain water 92 to 93%. Gases contain oxygen, carbon dioxide and nitrogen. In organic substances you can see the picture: plasma, carbohydrate, fats, amino acids, internal secretions, हार्मोन रिलीज़ करवाता है. नॉन इसेंशियल अमाइनो एसिड्स होते हैं, इसेंशियल अमाइनो एसिड्स होते हैं इसके अंदर एल्ब्यूमिन, ग्लोबुलिन, फाइब्रिनोजिन और ग्लूकोज. और ऑन द अदर हैंड इट विल enzymes, amylase, कार्बोनिक एनहाइड्रेस, एसिड फॉस्फेट, अल्कलाइन फॉस्फेट, लाइपेज, एस्ट्रेज, प्रोटेज, ट्रांसअमाइनेज, नॉन प्रोटीन नाइट्रोजन सब्सटेंस, अमोनिया, क्रिएटिनिन, ज़ियाथिन, हाइपोथिन, यूरिया, यूरिक एसिड. फंक्शन ऑफ ब्लड. Nutritive function, respiratory function, excretory functions, transport of hormone and enzyme, regulation of water balance, regulation of acid-base balance, regulation of body temperature, storage function, defensive function. Stage of cell development. Theories on blood cell formation. A monophyletic theory. First one is monophyletic theory. All blood cells come from one origin. Stem cells होमेटोब्लास्ट आरबीसीज, बेसिकली ब्लड सेल्स जो बनते हैं हमारी बोन के अंदर बनते हैं होमेटोब्लास्ट, इसकी पहली डिवीजन होती है बी थ्योरी, polyphyletic theory, also known as dualistic theory, which suggestive different group of blood cells originate from different stem cells, RBCs, WBCs, platelets, hemohistoblast, monocytes, lymphoblast, and plasma cells, tissue hemohistoblast. Stages of hematopoiesis. First one is mesoblastic stage, first month of embryonic life, when cells are formed outside the embryo in the mesenchyma of yolk sac. Hepatic stage by the 6th week, medullary stage by the 5th month. Blood cell formation occur in the bone marrow, primitive stem cells and committed progenitor cell are confined, spleen and lymph node, secondary lymph node tissue for lymphatic development and differentiation. Principle in normal cell maturation. Cell size. Immature red cells are much bigger than more mature cells. Cytoplasmic differentiation. Amount of cytoplasm, color, basophilia. Presence of granules WBCs गुजार करता है. Nuclear maturation. Presence, size, nucleus, cytoplasm ratio. Nucleus presence and lobes or indentation. Stage of erythropoiesis. Various stages between CFU colony forming units E cells and matured RBCs are pro-erythroblast, early normoblast, intermediate normoblast, late normoblast, reticulocyte, mature erythrocytes. Pro-erythroblast, megaloblast known as. Pro-erythroblast or megaloblast is the first cell derived from colony forming unit. It is very large in size with diameter of about 20 micro. Its nucleus is large and occupy the cell amount completely. The nucleus has two or more nuclei and reticular network. Pro-erythroblast does not contain hemoglobin. The cytoplasm in basophilic in nature. Pro-erythroblast multiplies several times and finally form the cell of next stage called erythroblast. Synthesis of hemoglobin start in the stage, however, appearance of hemoglobin occur only in intermediate normoblast. Early normoblast. The early normoblast is little smaller than pro-erythroblast with a diameter of about 50 micro. In the nucleus, the nuclei disappear. Condensation of chromatin network occur. The condensation network become dense. The cytoplasm is basophilic in nature, so this cell is also called basophilic erythroblast. This cell develop into next stage called intermediate normoblast. Intermediate normoblast contain cell is smaller than erythronormoblast, early erythronormoblast with a diameter of 10 to 12 micro. The nucleus is still present, but the chromatin network show further condensation. Nucleus present होता है लेकिन chromatin network further condensation ज़ाहिर करता है. Hemoglobin start appearing. Hemoglobin बनना शुरू हो जाती है. साइटोप्लाज्म इज अर्ली बेसोफिलिक, ऑलरेडी साइटोप्लाज्म ऑलरेडी बन चुका होता है. नाउ बिकॉज ऑफ प्रेजेंस ऑफ हीमोग्लोबिन की वजह से इट्स स्टेन विद बोथ एसिड एज वेल एज बेसिक स्टेन. क्योंकि हीमोग्लोबिन प्रेजेंट होती है इस वजह से ये एसिड और बेस दोनों स्टेन हो जाता है. सो दिस सेल इज कॉल्ड पॉलीक्रोमोफिलिक और पॉलीक्रोमेटिक एरिथ्रोब्लास्ट. दिस सेल डेवलप इनटू नेक्स्ट स्टेज कॉल्ड लेट नॉर्मोब्लास्ट. व्हाट इज लेट नॉर्मोब्लास्ट? In late normoblast, diameter of cell decrease further to about 8 to 10 micro. Nucleus become very small with very much condensed chromatin network and it is known as ink spot nucleus. Quantity of hemoglobin increases and the cytoplasm become almost acidophilic, so the cell is now called orthochromic erythroblast. In the first stage of late normoblast, just before it passes to next stage, the nucleus disintegrate and disappear. The process by which nucleus disappear is called pyconesis. Pyconesis is known as the disappear of nucleus. The final laminate is extruded from the cell. Late normoblast develop into the next stage called reticulocytes. Reticulocyte is otherwise known as immature RBCs. Reticulocytes is also known as immature RBCs. It is slightly larger than mature red blood cells. ये mature red blood cells से बड़े होते हैं. The cytoplasm contain the reticular network or reticulum, which is formed remnant for disintegrated organelle. Due to reticular network, this cell is called reticulocyte. The reticulum of reticulocytes stain with supravital staining. In newborn baby, reticulocyte count is 2% to 6% of RBCs, i.e. to the 6 reticulocyte presence of early 100 RBCs. The number of reticulocyte decrease during the first week after birth. Later, the reticulocyte count remain constant at or below 1% of RBC. The number increase whenever production and release of RBCs increases. Reticulocyte is basophilic due to presence of remnant of disintegrated Golgi apparatus, mitochondria and other organelle of cytoplasm. During this stage, the cell entered the blood capillaries through capillaries membrane from site of production by this epididymis. Matured erythroblast. Reticular network disappear and the cell become the mature RBCs and attain the biconcave shape. The cell decrease in size to 7.2 micro diameter. The mature RBC is with hemoglobin but without nucleus. It is required 7 days of development and maturation of RBCs from pro-erythroblast. It requires 5 days up to stage of reticulocytes. Reticulocytes take 2 more days to become the matured RBCs. Factor necessary for erythropoiesis. Development and maturation of erythrocytes require a variety of factors, which are classified into 3 categories: general factor, maturation factor, factor necessary for hemoglobin formation. General factor necessary for erythropoiesis are erythropoietin, thyroxine, hemopoietin, growth factor, vitamin B, C, D and E. Maturation factor, vitamin B12, intrinsic factor and folic acids are necessary for maturation of RBCs. Factor necessary for hemoglobin formation. Various materials are essential for formation of hemoglobin in the RBCs. Deficiency of these substances decrease the production of hemoglobin, leading to anemia. Such factors are first-class protein and amino acids. Iron necessary for formation of heme part of the hemoglobin. Copper necessary for absorption of iron from the gastrointestinal tract. Cobalt and nickel. These metals are essential for utilization of iron during hemoglobin formation. Vitamin C, riboflavin, nicotinic acid and pyridoxine are also essential for formation of hemoglobin. This is all about this. Please subscribe our channel and watch more videos.